All right, so we are ready. I mean, it is not too much past 10, right? Not too much past 10. 10:01. Oh my gosh, almost perfect. You know, so it is my great pleasure to welcome John Albeck, my colleague. I think it's the very, very first time in 20 years that I invited a colleague of mine to speak in our seminar, which is like, wow, inauguration kind of, right? So John Albeck is not just a cherished colleague. He at the Department of Molecular and Cellular Biology, which is actually my department for a reason that I still don't understand. And so he joined UC Davis in 2013. And before, he was did his PhD in computational and systems biology under Peter Sorger at MIT in 2007. And he was a postdoc and instructor in cell biology from 2007 to 2013 with Joan Brugger at Harvard Medical School. Oh, illustrious career. And he did a bachelor in Cornell in 2000. So his age index is 31, with his I-10 is 40. He has 8,000 citations so far. And today we will learn about biosensor imaging. So I think we are in for a treat. There will be beautiful pictures, I'm sure. You know, that's very beautiful. And you know, how these images tell us about individual cells and their bioenergetic states. John, the floor is yours.
All right. Well, thank you, Oliver, for that's a really kind introduction. And I did not realize I was the inaugural, intra faculty here. But yeah, it's an honor to be here. Thank you for inviting me. Yeah, and it's great having Oliver as a departmental colleague in the MCB. I'll say that probably I don't think either of us are competing for the attendance award at faculty meetings. So we have that in common. But anyway, it's a pleasure to be here. So let's talk about biosensors to begin with. If you're from Davis, obviously most of you are from Davis here, and you've seen me talk before. I have to apologize. You've probably seen these movies before. These were actually made about 10 years ago. And I just keep using them for the intro here. It's not that we haven't made new ones. We make hundreds of new ones like this every week, but we've never really made one that better shows what's going on with cells, right? And so let me give you a little tour through what we're looking at here. Number one, we are looking at ERK activity, right? And we're looking at a biosensor that tells us how active this kinase here, ERK, is inside the cell. And ERK, of course, has its own story. Oh, we're not seeing my little pointer, so let me see if I can get it here. ERK is right here. ERK has its own story. It's downstream of a number of growth factor receptors. It is typically activated when a growth factor like EGF binds to a receptor, first activates RAS, this kind of famous G protein, and then RAS initiates this MAP kinase cascade that ultimately terminates with ERK activity, at least as far as the kinases go. But then ERK goes on to phosphorylate many other proteins like transcription factors, cyclins, and it's generally required for the cell to go into the cell cycle. Usually if you take away EGF, a cell will simply exit the cell cycle and stay out of it for a while for most cells. And you can see that right here. So and the way we're watching it, I wanted to point out, was that we've built this little biosensor. It wasn't us who built it in the first place. That credit goes to various other labs in the world. But the idea is that there's two fluorescent proteins that have been engineered into this single peptide here. There is a site for ERK to phosphorylate, one of its favorite substrates. And there's also a phosphodieting domain, along with a little flexible linker here. And so when we get phosphorylation of this synthetic substrate by ERK inside the cell, that phosphorylation forces the binding of this WW domain that pulls the two fluorophores near each other, and then they can undergo FRET. We can read out the FRET by doing a ratio image of the CFP and the YFP, And that's what we're doing here. We've color coded that ratio since.
What does FRET stand for?
Oh, yes. Well, okay. Actually, I got a really interesting text on this just yesterday, right? A lot of people will say fluorescence, resonance, energy transfer, right? And what that means is basically when you're getting the two fluorophores near each other, and you excite one of them, you excite the donor, which is always the lower wavelength one because there's more energy going in. It will then transfer that excited electron state to the acceptor, which in this case is the YFP, right, the YPET. And it does this non-radiatively. It's kind of like a funky quantum effect. I don't think we fully understand it, but it requires very close proximity, right? So that's why anything it gets used a lot in biochemistry or molecular biology, because basically you have to get two things near each other on a molecular scale, right? The actual correct answer for FRET is Forster resonance energy transfer. I don't know the history on Forster, but my previous student, who's now at UCLA, and he's a first-year grad student there, he texted me yesterday. He said, "Hey, we're learning about biosensors. I didn't know Forster was a Nazi," and so, which was news to me. But I didn't get the backstory on that. So we decided maybe we'll normalize calling it fluorescence resonance energy, but we'll see. I don't know. The jury's out. We should do some deeper research on that exactly. So,
and on this image, is this the white cells here, or these, or what are we? Where is the FRET appearing?
Yeah. Okay. Yes. So the scale here. So the higher FRET is oranger colors, right? So you've got high ERK activity, and low ERK activity is white. And so you can see when we add the EGF, they suddenly turn orange, right? And so you suddenly get within minutes, within really 30 seconds or so, you get ERK active very quickly, right? And you can see subsequently, right, now there's lots more division happening, right? That within a few hours, that cycles up the division, as well as cell migration. There's a lot of other substrates of ERK we're not showing here that lead to cells changing their differentiation state, becoming more, sometimes becoming more mesenchymal, because you can switch from epithelial to mesenchymal states, and so on, right? And you can see them moving, right, and kind of undergoing more, just more dynamic activity in general, right? So we really got interested. These movies that really got us interested in kind of this little inner life of the cells, right? You can see, and now we've kind of transitioned to adding a little bit less EGF, right? The last time. Previous slide was like a bucket of EGF, which is normally what people do in biochemistry, right? They want to activate something, they put on more EGF than the cell could possibly respond to in 100 years, right? But this is a more physiological level of EGF, more like what you would see in the body. And actually, and it gets even more interesting when you do that, right? Because now the activities are very sporadic, right? You can watch one cell anywhere here, and you can see it kind of flip from the on state to the off state and so on. And we've spent quite a lot of time building software that will take this movie, find every cell, pull out every cell's little individual FRET trace, right? So you can get kind of you can get the individual ERK activities. If you look at the average of many of these, right, you get something very similar to what we usually see on a Western blot, which is you add EGF, ERK goes up a bit, it kind of drifts back down a little bit and stays high, and it just continues that way for hours. It looks from the mean like nothing is going on, right? But for the individual cell, lots of things are happening, right? It's flipping on and off, and in lots of different ways, right? The period of all these blinks is really quite different for every cell, although sometimes they can be very similar. Sister cells, it turns out, have a little more similarity to each other than to randomly chosen cells, right? So there is kind of a memory to this that's quite interesting. And so that's kind of spurred one of our, kind of the main themes of our lab, which is, can we understand what these different signaling programs mean for the cell, right? Do the different patterns cause the cell to go into different cell fates? And this idea it predates our lab quite a lot. If you go back to the 1990s, Chris Marshall was already talking about how sustained ERK could induce differentiation, and pulsatile ERK or transient ERK could induce proliferation. There's been a lot of talk over the years about how high levels of ERK seem to induce senescence. We don't really know what aspect of high it is. Is it an area under the curve? Is it amplitude? How high, how steep the peak actually is? Is it some combination of those? We don't really know, right? Even now, or even with all these biosensors, some of these questions have turned out to be quite elusive for the whole field. But nonetheless, right, there is that idea. And we think it's significant, right? Because if you look at different types of cancer cells, If you look at the average, you see kind of, like I said before, just, ERK goes up, ERK goes down. But if you look at the individual cells, you can see a lot of character that's different between the different types of mutations, right? Here we're looking at a cell, MCF7s. These are kind of a famous breast cancer cell line. They don't have a mutation in the ERK pathway. They've got some other things, but they still have this kind of sporadic pulses that you see similar to the MCF10As before, which weren't cancer cells. But if you look at these other two cancer cells, these ones that harbor a RAS mutation, right? This is kind of the infamous cancer RAS mutation here, A549 cells. These are lung cancer cells. These are also lung cancer cells, these HCC827s, but they harbor an EGFR mutation, right? So there's different molecular mutations here. You can pick up perhaps a little bit of difference in what their means are doing, right? But it's even more striking if you go down to the individual cell level, right? The RAS mutant cells tend to have ERK on solid, all the time, right? Not surprising, right? We know RAS is constitutively active, but they seem to reject all these other pulses, right? Something about this system basically sets it up so that they are just, they're flat. If you go to the EGFR mutations, right, it's back to being pretty dynamic. But if you look carefully at it, it's quite a lot more chaotic than these situations, than the non-mutant cells, right? There's not clear peaks and troughs. It's just kind of like a jumble all over the place. And we've seen that in multiple models when the EGFR itself is messed up. So this could be potentially pivotal for understanding how to treat these cells. We know that these cells behave quite differently clinically, right? They respond to different drugs, they have different prognoses. And so one of our questions is, can we, in the cancer world, can we now learn about these kinetics and then use that to better target what drugs we're giving to the patients, right? Is that something we could actually do? It's still a bit of a pipe dream, but but we're there. The way we think this all gets interpreted by the cell is largely through the lens of gene expression. Of course, that's not the whole story, but we've thought about it quite a lot that way, where ERK is going to activate, as I said before, many genes like transcription factors, other things, but it's going to do that by stabilizing certain transcription factors through phosphorylation, causing them to bind to promoters. It's also going to affect things like RNA splicing, RNA translation, protein degradation, RNA degradation. So it's got lots of inputs into that gene expression machinery. And so as a result, this might be the ERK activity that comes into this module here of gene expression. And this is actually a real cell where we tracked FRA activity. This is the output of this, right? So there's some kind of connection between these that we don't fully understand. And a number of genes, famous ones that are downstream of ERK, like MYC, FRA1, FOS, they all kind of follow the same sort of multi-regulation pattern, right? So we think that it's important to figure out that code. Okay, so
by the way, there's a lot of chemists here.
Yeah.
So they have never heard of ERK or EGRF.
Excellent.
Right? So or RAS. I mean, you're not speaking to your peers.
Okay.
Speaking to, well, some of them.
Yeah.
Like Joanna and whatnot, but not everybody knows the basics here.
Yeah. No, and it's fine. Actually, that's I'm simply on a similar point in my career where I'm trying to learn more chemistry, right? I was a biochemist. I sort of left a lot of it behind to get into this molecular world. But now I think, as I'll explain in a minute, kind of the getting into the metabolism has kind of pulled us back here, right? So back to that. So I'll hopefully get to that soon. I don't want to take too long on my little chunk here, because my goal for the first section here is really just to kind of get a sense of what we can do and what kinds of things we like to think about. And then hopefully within a few minutes, I'll hopefully be into the metabolism. So go ahead.
From what I understand, all of these are the downstream for ERK. The ERK was kind of expressed all over the cell, whereas this is DAPI and ERK, EGR1 is all on the nucleus.
Yeah, they're all very nuclear. Yeah, that's a great point. Yeah, ERK is more or less a soluble protein. So it can basically be anywhere in the cytoplasm or in the nucleus. It does transit between the nucleus and the cytoplasm. And for a long time, people equated that with activity, but it turned out not to really be strictly true, right? It's very much true that ERK could phosphorylate something in the cytoplasm and that substrate could move to the nucleus, right? And so you can't necessarily take too much information from where ERK is because it's moving around constantly, and so are its substrates. But these substrates that you see, they are pretty strictly nuclear proteins. Although that doesn't mean, this goes back to chemistry, right? It's an equilibrium, right? They spend most of their time in the nucleus, and that's why when you stain for the protein with an antibody, you see mostly nucleus. But it doesn't mean they don't leave occasionally to go to the cytoplasm through the nuclear pores. get phosphorylated there, and then come back, right? So, and in fact, people have proven that that's true. You can do FRAP, and there's quite a lot of motion between the different cellular compartments. So sometimes what we see just by localization doesn't do justice to what's all going inside, right? There's a dynamic equilibrium of virtually every reaction you can think of, all those steps in the pathway, right? And so it gets complicated. But the point that we wanted to, just to sort of wrap up our kind of idea that ERK can control things based on its dynamics. If we do look at the overall expression level of these two different proteins, FRA1 and EGR1, you can see that they respond to different patterns of ERK activity, right? So, and that comes out of, we can show that it's true out of the chemical equations, right? We can build a differential equation model like this one here, where we represent each of the phosphorylation events and the degradation events and so on, and the production events as actual differential equations, right? And we have rate constants associated with them. If we build that network, we can get the same kind of behaviors. And to get the two different genes to behave differently, what you realize you have to do is that you have to change the parameters of their activation, right? And so EGR has different parameters than ERK. EGR has different parameters than FRA1, and that means that when ERK turns on, and we're looking at ERK in blue down here, right? And what we did was we gave the model actual ERK traces and said, okay, predict what the two different genes are going to do. If we gave it this pulsatile pattern of ERK, EGR1 tracks with ERK pretty closely, right? Because of its rate constants allow it to degrade rapidly, and so it can track very closely, whereas FRA1 is much slower in its regulation. And so it can build up over time. It really ends up integrating all these pulses of ERK activity over time, right? So these different readouts downstream of ERK can interpret the dynamics, right? And they can respond differently depending on what genes they activate, right? Both of these, FRA1 and EGR1, are transcription factors. And so depending on what kinetics ERK has, you're driving different transcription factors, and then therefore different sets of downstream genes, and that can make a cell do different things, right? So that's one of the ways we've thought about this and dealt with it. And another way is to do a completely agnostic model, right? Here we built into the model all the chemistry that the biochemists have worked out over the years, what binds to what degrades what, and so on. Here you can do it, sort of the more modern AI data-driven kind of way, where you just take lots and lots of data, right? All those little individual traces. We can break them down into different characteristics like frequency or amplitude, or we can even just throw all the time points just raw straight into the model these days, and ask it to predict something like how much FRA or EGR1 comes out of the model, right? Comes out of the system, and that works too, right? Ram, and I'll just shout out Ram here, who is a grad student in the lab up until last year. He did both he worked as an undergraduate and a junior specialist and a grad student in the lab. Eventually, we had to kick him out, but not before he succeeded in setting the record for the amount of data collected, which was, in this case, this is just one of his early experiments, 97,000 cells. Every line here is one cell. And what Ram did to really try to decode this whole system was to first track live ERK activity, just like we did a minute ago, like in the movies. And that's what you see on the left panel here with the blue and the yellow. The yellow is active ERK, blue is inactive, and every line is one cell. And you can kind of see the little blips of ERK activity that every cell has. But then what he did was he fixed the cells at the end and then went through a series of immunofluorescence stains for the different ERK targets and various other things that he thought might be related to ERK, right? And so he built up this other library of stains, and it was all registered to the same cells, right? So he took the same plate, the live image, fixed it, stained it, took it back to the microscope, took more images, washed it off, fixed and stained it more, and kept doing this every time, collecting the same cells worth of data, right? So now we have this kind of Rosetta Stone where we have both the activity of ERK when the cell was alive, and then at least at the time it was fixed, how much of these different proteins were expressed, which ones were phosphorylated, and so on, right? And so Ram spent a lot of time building this data set, and then also worked with Gerald Kwan and other people in the department to develop models of this, right? And I won't go through all the models because Ram, he tried lots of different things. In this case, he tried to teach a convolutional neural network, or basically a machine learning algorithm, how to predict each of those different stains, right, off of just the live data, right? He said, can I take the live data, teach the model what stains, which proteins turn on, with this model and then use it to predict things. And it worked out fairly well, right? The model did as good a job of as any model we've seen at the single cell level. That means often that it's single cell models aren't great. You know, if you're getting close to 0.6, that's close to the best single cell model for that we've seen here, right? But or that other people have seen. But nonetheless, the convolutional neural network does a pretty good job of learning that. Sometimes it does much better than like a simple linear model, and sometimes it doesn't do much better than the linear model. And there's insights to be gained there. But
so the missing point to having one R square of one is 40%, right?
Yeah, so, exactly. Yeah.
So what do we think about it? Why can't we explain?
Why can't we do better? Yeah, that's a great question. I mean, there's this. Part of it's probably measurement noise, right? You know, I don't think we're not accurately measuring everything exactly, right? Part of it is probably just, you know, we're missing kinetics, right? You know, this is we're already using a single time point, right? And so we're not getting all the kinetics there. There could be. And the other thing is, other things besides ERK could be affecting each of these things, right? There's independent regulation, right? So, yeah, if you look, so Lucas Pelkmans at Zurich he's done a similar kind of experiment, but just looking at the RNA for a given gene and then the protein for a given gene in the same cell, right? And they also come up with R squareds in the 0.6 at best 0.7 kind of range, right? They're really, you know, even for the same gene, right? So it makes you realize that even for two things that we think are intricately connected, there's a lot of other things that you can't pick up just from how much RNA is there, right? Other things like how many ribosomes there are could matter, right? And so, or where the RNA was, you know, sequestered, you know, inside the cell. There's things that we're not picking up in our data that are still important factors, and that factors into the into that. So, so the one application of this I'll kind of highlight that RAM came up with is that you can run the model in reverse. And you can then take your fixed stains, right, and then predict what kind of kinetics the cell had, right? Because we've got that data set, that Rosetta Stone, you can kind of go back and forth. He showed that you could do this, right? That you could predict reasonably well what kind of kinetics a cell had just based on the stains alone, right? And we think that might be potentially important, right? Because if you, going back to, like we said, we wanted to understand how these different kinetics might be important in cancer, and do they impact how a patient's tumor cells respond to a particular drug. We're never really going to be able to do biosensors in a patient, right? You know, we can't engineer it into the genome, you know, the biosensor into the genome of the patients. Getting it into the cells after you've, you know, biopsied them out of the patient is relatively difficult, but it's probably doable, but it's not going to be something we do on a routine basis to test a patient. But immunohistochemistry, immunofluorescence is, right? We do this constantly. This is part of the normal workflow. You get a biopsy, it comes out, the pathologist puts it through a round of stains, Ki-67, all these other things that are standbys, and they use that to assess what the tumor is doing. What we realize is that, you know, with Ram's model, now we can add on a few of these stains, FRA1, EGR1, MYC, and so on. And his model can now tell us what was happening inside the tumor before the biopsy got punched out, right? And before you can, and so what was happening kinetically, right? So that's work we're kind of pursuing now with Jonathan Rees and Surabhi Singhal over at the Cancer Center, right? We're actually trying to do this on actual patient samples or patient-derived xenografts and so on. So, you know, the jury's out whether we can really do this outside the dish, right? This works well in a dish. Does it work in, you know, in little histochemistry slides? You know, we'll find out. It's certainly going to be a little challenging, right? But it's something we're working on. Okay, yeah, and then here's Ram's stats on how well those models doing and so on, right? But okay, like I said, I wanted to get to some metabolism today since this is the metabolomics seminar. And so, yeah, I suppose it's not a surprise to you, especially the people who haven't heard of ERK, it's not all about ERK, right? There's a lot more to what the cell is doing inside its life, its little internal life, than just ERK, right? And of course, one of ERK's closest friends and neighbors, right, it's talked about with it very frequently, is this PI3 kinase AKT pathway, which you probably can't help but notice is the pathway downstream of insulin, which obviously is a metabolic driver, right? You know, and we eat food, insulin gets secreted into our bloodstream. It tells all the cells what they should be doing. They should take up glucose. They should activate various biosynthetic pathways. And there's a lot, we've known for years that there's lots of crosstalk between the ERK pathway and the AKT pathway. And of course, downstream of both of them, you've got good old mTOR, right? This, you know, famous kind of regulator of protein synthesis. And you've got joint regulation of various genes, right? So there's a lot going on there just to begin with, right? But then, I think, what led us into the metabolism world really was the realization that we can't just stop there, right? We do have to think about the real mainstay of the cell, right? Glycolysis, the Krebs cycle, all these things that really make the cell run and they make every cell run, going back to bacteria, right? This is, life, right? And I think one of the things we forget in signal transduction a lot is that we learned about all this stuff here, all these kinases, because we started with metabolic enzymes, right? And it turned out there, Ed Krebs, who worked over in Tupper for, quite a while, right? And discovered protein phosphorylation, right? That's kind of one of my favorite fun facts about UC Davis, right? Protein phosphorylation, like the guy who actually discovered that proteins get phosphorylated worked right over there, right? Which is pretty amazing, right? He didn't discover the phosphorylation here, unfortunately, right? But he did work out a lot of this cool stuff here, right? And what they realized is that the phosphorylation, they were looking at metabolic enzymes and trying to understand how they were switched on and off, right? And it turned out it was phosphorylation, and that led. And they said, well, what things are causing the phosphorylation? And that turned out to be these various kinases, right? And PKA, PKB, PKC, and so on. And so the kinases started coming out their ears, and they were, and we made all these. We learned them that they were involved in various cascades and so on. But at the end of the day, right? That's probably one of the major things these are doing, right? Is controlling how metabolism works, right? All the basics, glucose intake, amino acids into the TCA cycle, production of ATP, right? And there are some very key kinases like AMP kinase that are sensors for how this pathway is working, right? AMP kinase senses how much ATP there is in the cell, or really the ratio of ATP to ADP and AMP. In other words, the energy charge, so how energetically charged the cell is. And it talks directly to ERK, through the MAP kinase cascade. It talks to mTOR. It talks to the AKT pathway. It controls gene expression and so on. So this whole thing is really you can't really pull it apart, right? It's all one big network. And so really, our vision has been not to think about just ERK, but with the biosensors, right? Here's our good old ERK biosensor that we saw before. But can we use similar biosensors for things like AMPK to watch what's going on metabolically? And they so far they haven't disappointed, right? If anything, they can be more wild than the ERK activity, right? Here we managed to induce a little dance party with by adding a glycolysis inhibitor of GAPDH. Basically, just seems to cross some important wire somewhere, right? And then the cells go into this kind of well, it's basically a terminal oscillation, right? They're going to do this for about a day or so. Every 30 minutes, they cycle on and off, and then they just die. They can't really handle. Good. Yeah, exactly. Great question, right? And actually, perfect segue to the next slide, right? There are quite a lot of sensors built in the world, right? And most of these are built by other labs, and it's a really fantastic community. People share biosensors right and left, with very little, worries about MTAs or intellectual property. It's a very great community, and the answer is sometimes, right? And if there's different ways to do it. The easiest way, of course, is different colors. And there are different colors, and some of some very clever, a very clever postdoc at Stanford, Sergi Vergo, has now got his own lab at Johns Hopkins for quite a while, came up with this idea that you could look at the translocation of certain proteins between the nucleus and the cytoplasm as a readout of kinase activity, right? Because kinases will often regulate certain proteins, and you can sort of take advantage of this. And we have to make an mTOR sensor, for example, or an AKT sensor and so on. And very often when you're doing this, right, you can switch the color to whatever you want, right? So what's normally possible is 3 is easy. 3 is no problem, right? You can easily do a FRET sensor, CFE, YFE, plus an orange translocation, plus a red translocation. 4 is probably not so bad either. You could just, now that there's far red proteins, you could do a far red translocation, right? And you'd be doing pretty good. But not everything has a translocation equivalent, right? There's a lot of things that are still stuck in Fretland, right? Or something similar, or this circularly permuted fluorescent proteins. They were built based on these very, color specific frameworks. And so we can't change them very easily. We can't just switch it to a red. It doesn't work that way, right? So the reds don't have enough of a structural overlap with the greens that you can just, swap them in and out for these very sensitive biosensors that are often based on small conformational changes. So yeah, it's a very good question. So yeah, and so we have started multiplexing them, and I'll come back to some of that data in a second, and some other ideas there. But all that heterogeneity makes us think that, okay, if we're seeing it at the signaling level, right, and it's likely to be happening at the metabolic level, right? It's, metabolism is a very regulated thing, right? We know that's a crucial aspect of how our body works, and you could argue it's the most immediately crucial, right? If metabolism breaks down, you are in trouble quickly. And we know in cancer that it is very frequently corrupted, right? A normal cell will usually use a variety of fuels, right? Glucose, amino acids, then we'll go into the TCA cycle. Much of the ATP is being generated via oxidative phosphorylation. In cancer cells, often something goes wrong. In the case of these renal cell carcinomas, which are this type of cancer that we started working on thanks to a collaboration with Shuchi Gulati over at the Cancer Center. In these cells, very often they have a disruption of a protein, von Hippel-Lindau, VHL, that controls this transcription factor known as HIF. Are we coming closer to what people know? Who knows about HIF alpha? Anybody? Okay. Oh, only John. Okay. Oh, and a few others. And so I'll explain quickly. So HIF alpha is another transcription factor. This one is regulated by, as you might expect, oxygen, right? And it typically turns on when cells are low in oxygen. Bill Kalin, who's actually going to speak at the Cancer Center, I think maybe in early 2026, got the Nobel Prize for working on it. And it's this really crucial transcription factor. It turns out it's not only regulated by oxygen. There's other things that can control it. And typically when these cancer cells lose VHL, HIF1 alpha becomes hyperactive, turns on many genes, and it does the things that a cell might want to do when it's low on oxygen, i.e. it turns on glycolysis, right? Rather than using oxidative phosphorylation, which requires oxygen, the cell now is able to do much more import of glucose, burning of glucose, and secreting of lactate, which is kind of this, like it's essentially fermentation, right? It's less efficient than oxidative phosphorylation. It produces lactate, which is not always a desirable byproduct, at least in high concentrations. But these cancer cells love to do it, right? And this is why cancer cells, if you ever do it, who knows what an FDG PET is, right? Okay, no? Okay, good. So if you have cancer, right, and the doctors are worried about where it's metastasized to or whether it's metastasized, you'll go for a PET scan where they will feed you a radioactive analog of glucose, fluorodeoxyglucose, right? And that FDG, fluorodeoxyglucose, gets cancer cells imported at a much higher rate, typically, right? Because they've usually gone through some kind of change in their metabolism that makes them want to do more glycolysis, and so that radioactive glucose will build up inside the cancer cells because it's deoxy, so it's engineered so it can't go through the whole glycolytic cascade. It kind of gets stuck, right? So it builds up, and you get these dark radioactive spots when you scan, if you scan the positron emissions from the body, right? And Davis is actually, another unique thing about Davis is they built the the PET Explorer, which does other kinds of PET, but it can do FDG much more sensitively, right? Because they've built a much bigger detector ring than others, right? So there's a lot of exciting things happening in this area, understanding how metabolism leads to cancer and it affects how cancer works, and it also allows us to perhaps to detect and perhaps treat cancer. So that's what happens in cancer. You know that the cells are becoming more glycolytic, but our question was, okay, that's a fairly striking change, right? We know that sometimes it also happens in normal cells. Like a T cell becomes activated and it usually switches to more glycolytic metabolism as well, because that higher rate of glucose burning can support the protein synthesis and the cell division that the T cell needs to undergo to replicate and kind of attack the tumor, right? So, or attack whatever threat that it's trying to attack. It's a T cell, right? And so T cells are metabolically regulated, right? So there's a lot of things to make us think that really this switch is something we should look at. And when I first got into metabolism, I think early when I came here to Davis, I was really excited that Oliver was here and had this world-renowned West Coast Metabolic Center right here, right? People, I've heard people talk about it other places, right? This is a very very well-known place, right? And I was like, great, they're just going to we're going to go over, we're going to get a metabolic scan of our cells, and and that's just going to tell us whether they're glycolytic or oxidative, right? And then Oliver had to explain to me, no, it's really not quite so simple, right? You can't just catalog all the metabolites, right, and then know what's happening, right? There's a lot more subtlety here, right? And a lot of it comes down to bioenergetics and potentials and actual fluxes, right? And so it's really far from trivial, right, to actually assess what a cell is doing, oxidative phosphorylation versus glycolysis, or many of the other things outside that, right? I mean, I think the WCMC has done, amazing things, finding all these other metabolites. We're still kind of stuck in the little, in the middle, the little core here, right? Trying to understand how it's switching, right? But there's of course lots of other things going on. So I'm not doing a total justice to metabolism here by representing it like this, right? But if you do represent it like this, right, where you have your ATP being produced by oxphos and glycolysis at perhaps at different rates and perhaps at variable rates between the cells, then and then used, right? So the usage basically cycles the ATP back to ADP, which then has to get recycled back to ATP and so on. This whole process is happening in large part thanks to oxygen working through oxphos. If you do want to understand how that's working, one of the classic ways to do this is the seahorse assay, right? Which is basically where you're tracking both oxygen consumption rates and lactate secretion or pH in the same culture, right? And if you do that, actually, you can at least for a population of cells get a pretty good read on how much they're using oxphos versus glycolysis to make their ATP, right? This is pretty, this is pretty well worked out. You can just buy kits. There's a core facility over in VetMed that has a machine. You can just run them through that and get your calculations. That's all great. But that tells you about 1,000 or 1,000,000 cells, right? It doesn't tell you about individual cells and what's different between them, right? And I just want to point out that one of the key things you do here is first to treat the cells with oligomycin, which knocks out their ATP production from oxphos. And it also, at the same time, causes them to stop using oxygen, right? Because they can't keep using oxygen if they're forced to stop making ATP that direction. So now you're only making ATP with glycolysis in that case. That gives you this little drop here, right? Which basically tells you what your ATP production from oxphos was. I'm just going to pointing it out because we realized that if we were going to do this at the single cell level, right, we would need some way of detecting that shift upon oligomycin in individual cells, right? And there aren't really good biosensors for oxygen, right? You don't have like an oxygen biosensor that works very reliably, right? There's some progress in that area. I don't want to undercut it, but it's not really ready for general use by by people like us who aren't actually that good at that sort of things. But anyway, but what we could do is we could look at AMPK activity, right? Remember, AMPK is sensing that energy charge, right? That is certainly going to shift when we treat the cells with oligomycin, right? And so if a threat detector can tell us about AMPK activity, then perhaps it can tell us how much oxphos the cell was actually using. And with the idea being that, okay, if cells are using some combination of glycolysis and oxphos to fill up their ATP tank, and then the usage is kind of depleting it, you could have cells with similar high levels of ATP, similar high energy charges, low ADP, AMPK is off everywhere, right? But if you now treat them with an oxphos inhibitor, knock out that one key arm, now they're potentially imbalanced, right? Cells that are not able to do a lot of glycolysis to keep up with their usage should now have an increase in ADP, and that should trigger AMPK activity, right? Whereas a cell that does have plenty of glycolytic capacity and perhaps not so much usage would still be fine, right? It's still capable of meeting its needs. You don't fill up your ADP tank, you don't turn on AMPK. So that was kind of our reasoning for how this might work. And Nott and Brienne went through and did this actual experiment. They set up MCF10A cells here with a biosensor for AMP kinase activity, right? So they're setting up this exact experiment that we had shown before. They then treated the cells with oligomycin that knocked out the ATP production from the mitochondria. And you got something like this, right? Right there is the oligomycin, right? That's turning on AMPK very rapidly. And like I said, these biosensors haven't disappointed. There's a ton of heterogeneity here, right? We thought we might see something like this, and sure enough, we did, right? And if you look carefully at some of the single cell traces, you can see that at that initial point, right, some of the cells had a very big response. AMPK turned on very rapidly, and other cells had almost minimal response. Of course, there's a lot more complications to it, right? There's the cycle of every 3 hours it turned on and off, even though we've kept oligomycin there the entire time, right? These cells are running purely on glycolysis, and it seems like when they do that, they have to go through these periodic cycles of AMPK turning on and turning off and so on. So there's, it not only told us that yes, there's heterogeneity, but there's also a lot more going on under the hood than we actually realized. From the standpoint of actually how do we catalog this heterogeneity, and how do we take a population of cells and tell you, who's OXPHOS and who's not, we realized that we could simply just take that first peak and quantify the area under the curve of it. The cells that are the non-responders, in other words, they didn't need OXPHOS to keep their energy charge low, or high rather, and they didn't trigger AMPK activity, they would look like this. And then the cells that did feel the perturbation because they did require the OXPHOS, they would have a much bigger perturbation. So we can do that, we can quantify that, we can go across a whole population and make histograms and see that, yeah, very often there's this subpopulation of cells that has that has apparently not very much need for OXPHOS, right? These are the non-responders. They are what we call OXPHOS independent cells. And then there's a much larger population of OXPHOS dependent cells. If you go across many different types of cells, you can look at how heterogeneous they are and also what their set points are. Our MCF10As over here, you can see that, if we actually feed them lots of insulin, really turn on glycolysis, glucose uptake, they're fairly evenly split. Some of the cells are relying on OXPHOS, some are not. And as we take away their insulin, right, they become much more dependent on OXPHOS, as you would expect. But there's other cell types that are very glycolytically capable, right? Very few OXPHOS dependent cells. And once you feed them insulin, they're essentially all OXPHOS independent. Some other cells are more OXPHOS dependent, like MCF7s, no matter how much insulin you give them, right? So you can actually now, we think this kind of basically is a little bit like a single cell seahorse. It's not perfect, right? But it is giving us that heterogeneity between cells. There are other ways to do this, and so we wanted to kind of but yeah, so one thing we want to do is go back to that classic model, right? Those the 7860 cells, the renal carcinomas that I mentioned before.
Okay, so
yeah.
if I may interject again.
Yeah, of course. Yeah.
So why are individual cells indivisible?
Yeah, that's a great question.
I mean, you have in principle the same genes, right? And there's some mutation somewhere, right? Okay, good. And now you multiply them; they still have the same genome.
Yeah.
And then they express individuality.
Yeah, exactly.
Some are like, no, I'm like whatever. Yeah. I'm, and then we are different. So why would they? How can they choose?
I think the short answer is, I mean, hopefully this room will like this, but I don't think the genes are in charge, right?
Oh, okay.
It's metabolism that's in charge.
But also the media. I'm a big fan of environment and media. But they have the same media.
Yeah. But they have lots of, as they go through the cell, so okay, I think the first thing that comes up often is it cell cycle, right? It's weakly linked to cell cycle, right? But not strongly, right? If you look at an early G1, it tends to be more OXPHOS independent, right? And then as you go through the cell cycle and you have more protein synthesis needs, you tend to be more OXPHOS dependent, right? Because you need lots of energy, right? But it's a weak effect, right? I think there also seems to be a metabolic cycle of the cells, right? This 3-hour period that we see very distinctly when we add oligomycin, you can still pick it up without oligomycin, right? It's still there, it's just a lot weaker, right? And I think cells go through this cycle, right? And what we know is cycling along with AMPK, right? So AMPK, which is telling us that the AMPK is on or off, and the energy charge is going up and down. Other things like mTOR activity or ERK activity will cycle with it, right? Oppositely, right? And so it seems like the cell, so if you think about it, things like autophagy, right? You don't want to necessarily be making more proteins like at full speed, like mTOR's on, right? And doing autophagy, like just breaking down proteins, right? It would make more sense if if the cell kind of traded off between these different phases, right? Joanna, go ahead.
So cycles always 3 hours. So this is in cell culture.
This cycle we see here is more or less always 3 hours. It doesn't matter which electron transport chain inhibitor we use.
Is it going to be different? Not 3 hours if it's actually in vivo,
Yeah, I would like. So we haven't got to the point where we can see it in vivo yet, right? And so we're hopefully soon. So what we've done recently with the help of the Mouse Biology Project is to engineer a triple biosensor mouse. We call it T-Cameo. which basically stands for a TFEB reporter, like the mTOR one I mentioned earlier, an ERK reporter, and the AMPK reporter, all under one promoter, right? And so now we can express that in various types of cells, right? And so, but we just got the mice over the summer, basically. And so now we're at the point of, oh, great, there's a fuzzy thing that we don't work with normally sitting on our bench. How are we going to?
I wonder if it's linked to eating fasting. Could it be more than 3 hours?
Yeah, that's a really good question. Like, I mean, there's something, I'm not sure if I kept these slides in. I apparently didn't, right? But like it's induced by different drugs, but for example, metformin, right, which is a class, a diabetes drug that we've been using for 50 years more. We know that it needs a weak ETC poison. It poisons complex 1, possibly complex 4, and causes cells to take up more glucose, which is why it works well for diabetes. It will induce the same 3-hour cycle, right? Or at least amplify. Maybe the right way to say it, I shouldn't say induce, maybe amplify, right? Because like I said, we see the cells having this kind of weak cycle, and then when you sort of constrain them, they force them to do one thing. They really kind of amplifies that cycle. And like I said before, the other kinases kind of go along with it, right? So I this is still a mystery to us. I don't know exactly. Yeah.
Is there any evidence of like crosstalk between the cells that could be coordinating the cyclical part of it?
Yeah, between the adjacent cells or cells. Yeah, I think so. I mean, although we have fairly weak evidence for it. I mean, if you look at those movies, that you see some spatial correlation. We haven't really played around with cell density very much yet. We probably really ought to. But that's not something we've looked at carefully, right? So I think the answer is, and of course, if cells had sort of tight junctions and you could actually, move metabolites through them, intracellularly, right? That would be, a clear case where they'd be linked, and these cells don't have those, right? So I think there's definitely a lot of potential for that, but we don't have a firm answer. It's a good question, though. Yeah. All right, any other questions? We're getting close to the end, so I'm gonna. I first got way too many slides, but I will hopefully find a way to sort of wrap them up in the next couple minutes here. This is kind of modeling what causes the cells to using the same kind of models we'd used before, like the data-driven models, to ask the cell cycle or the expression of a major glucose transporter or the amount of protein synthesis in a cell. Do these predict whether the cell is going to respond to oligomycin with AMPK activity? Does it tell us between the OX-phos dependent versus independent? And we can build an okay model. It's a really bad model, right? So far, I guess in the paper we had to call it an okay model, but really it's a terrible model. Our percent variance explained, R squared in other words, is less than 10, right? We are not doing well here. Right, and so I don't think we have the full answer. Certainly, the cell cycle gives us a signal, right, on its own, right? The GLUT1 expression gives us the most contribution, right? But there's got to be a lot more going on here under the hood that we don't.
If you don't use a PSR model, but some kind of more,
alignment. Yeah, that's fair. That's fair.
Some kind of transformers.
Yeah, exactly. A nonlinear. These are likely nonlinear effects. We should probably be using nonlinear models, right? So that's a good point. Yeah. So I guess I'll sort of wrap up with some recent work by Christy Abate, right? Who, because we think glycolysis is kind of one of the main things that's being different between these cells. Fortunately, a group up at OHSU, Dick Goodman's group, had developed a very nice glycolysis reporter, we'll call it that. Really what it is it's a reporter for fructose 1,6-bisphosphate. And so it senses this very central intermediate in glycolysis, which is thought to be, depending how you ask a biochemist, they'll give you different answers, but rate limiting, rate related. It's the first, irreversible step in glycolysis and so on. And nonetheless, it tells you something about how much glycolysis the cell is doing. Here's a movie that Christy made, really, and perhaps this is in the running for the one to replace the first movie in my talk, right? Because it's really, really quite nice. What she did here was she first starved the cells. In this case, we're looking at a scale of purple, meaning low FBP, up through blue. She likes the rainbows, and they work well. You'll see. Blue meaning high FBP, right? And so here she's starved them first, so they're at low FBP, and now she's going to add glucose, and suddenly they all taking glucose, right? And it turns green, goes way up the scale, right? And then it kind of goes back down, right? And then the heterogeneity shows up, right? Some are pink, some are purple, some are orange, some are green. They seem to be, at the very least, we can say they have different levels of FBP, right? Which implies that they're likely doing different glycolysis at very different rates, right? And so Christy really got interested in this.
What's the timescale?
Oh yeah. This is too new to have a timescale on it. The whole movie is about 1 day, right? So and we're taking an image every 6 minutes. Here's some quantification of what it looks like. That very first peak when you add glucose takes place in about 5 or 10 minutes. If you blow it up here, here's that first peak, right? And you can see cells actually, even at that first peak, have some very different behaviors. Some take in a ton of glucose. They were, it's like you opened up the door to the snack bar and some cells just ran in and immediately ate an entire box of Snickers, right? Other cells were a little more judicious and actually you're like, no, I'm going to wait for later, right? So there's quite a lot of individuality even there, right? And then there's a second peak, right? Also has a fair amount of variation that I'm not showing here. Oh, I guess here it is. Right? Here's that second peak. We've now compressed the scale, so the first one is just a little blip on the dotted line. But the second peak, also different levels. And then there's a third phase afterwards where cells kind of go through, again, kind of this three-ish hour period that pops up again, right? Of FBP going up, FBP going down. Is this really glycolysis going faster or going slower? We thought so at first, but then we realized that we can't think so simplistically, right? Christy started doing some experiments with things that perturbed the transition from glycolysis to the mitochondria. In other words, when pyruvate comes out at the end of glycolysis, it has to go to, or it doesn't say have to, but one of the fates is it can go to the mitochondria, become oxidized through the TCA cycle, make ATP. But if you perturb how much pyruvate can go into the mitochondria using an inhibitor that blocks the transport, we expected initially, right, that we were going to, we thought that was really going to speed up the cells more FBP, right? Because we're like, okay, well, if the cell can't do, if it can't do oxphos, right, because you're not letting pyruvate in, it should be doing more glycolysis, right? And so FBP should go up. And we saw the exact opposite, right? If you look right here, the green line is the pyruvate carrier inhibitor, and unlike so the control was red, right? The pyruvate carrier inhibitor drops it down immediately. Oligomycin, which also blocks the ET cycle, makes it go up, and this just seemed completely wonky. We couldn't understand why this would be the case until we realized that we were missing a really key factor, which is the NAD plus and the NADH, right? And we realized that the step here below FBP, in fact, the GAP-D8 catalyzed step, requires NAD plus. And if you cycle too much of your NAD plus to NADH, you have no more NAD plus to run that reaction anymore, right? So you end up bottlenecking the reaction. And that's exactly what Christy was seeing. And she's able to prove it by basically pre-treating the cells with either pyruvate or lactate. Pyruvate goes in, forces more NAD plus to be made, and basically completely wipes out your FBP signal, right? Because basically what you've done is open the gate below FBP, so that pipe is very wide and you can never build up enough FBP to really see it. Lactate does the opposite. Even though they're energetically more or less equivalent, lactate is going to force the LDH reaction here towards more NADH, right? And so, sorry, more NADH. Oh, and you can't even see the pointer that I'm trying to point. But so you get more NADH if you add more lactate, right? You're forcing the reaction this way. When you do that, now your FBP signal upon glucose addition is huge, right? So it's really, we can't just think about FBP, right? It's one data point, but it's not interpretable unless we know what's going on with NADH and NAD plus, right? And that really comes back to this whole multiplexing question that you brought up before, is how are we going to do this? And it turns out there are some really good NADH, NAD plus reporters, but they are the exact same color as the FBP reporter, right? So how are we going to solve this problem, right? So that's kind of where we're at right now. So Christy's trying to solve. We're almost out of time, so I'm just going to skip over the last bits here. Lots of bits, right? I never, but no, we had. I really appreciate the conversation that we can have. Much more so than being able to get through the whole story. Because I think I've told you the important parts here. So I just want to thank the people in the lab who did all this, right? And you can see Christy and Ram and everybody all in this picture here. I should have somewhere there was a. This one is not annotated with who the people are, but this is the really fantastic crew that's been in the lab the last couple of years. And our funding and our collaborators. And so I'll take any more questions if you have them. And thanks for your time.
Thank you. All right. So, Shen.
When there's probe or sensor for small molecules,
Mhm.
BPs,
Yeah.
also 1,6 and 1,26. How
Yeah.
about the specificity?
Yeah, often so okay. The way those are usually constructed is you take the circular permuted GFP, and when circular permuted GFP is basically it's GFP except you're making the same beta barrel, 11-stranded beta barrel, but you can basically move the order of as long as you keep the same cycle of 1 through 11, you can put 6 through 11 before 1 through 5, and it still folds the same way. It still folds into the same barrel, right? And when you do this, it makes the N and C termini, certain permutations make the N and C termini very sensitive. And so what people will do is they'll take a known allosteric binder protein to the small molecule you're interested in. Often they'll go fishing in bacteria to find something that binds NADH or binds FPP. And that's in both cases, that's what they've done. They've taken those proteins, stitched them onto the circular permuted GFP such that that little conformational change you get from binding the metabolite now deforms the N and C termini and basically changes the fluorescence, right? So, so your question was how selective they are. It depends on what that binding protein is, how selective it is, and whether it undergoes a conformational. Because often these things will bind to other metabolites, right? In the case of highlight, it's very likely that it is sensing other hexoses. Similar. There's a good chance that it's not being super specific. For our purposes, that's okay, right? Because really we're using it effectively as like an upper glycolysis pool sensor, right? And then, really the NADH is telling us about, we think, the lower end of the pool, right? But, yeah, they're not always perfect, right? And so you have to really factor that into what you're thinking about. Yeah, over here.
Yeah. Going back to the heterogeneity, is that sort of behavior typical in healthy tissue, or is it like it's meant to be a hallmark, like a diagnostic point for?
Yeah, that's a great question. I don't think we know yet, right? I do think that healthy cells are very heterogeneous, right? But I think what we think is more and more is that very likely they have some structure to the heterogeneity, right? When people have looked at ERK activity in endogenous tissues, it's very often in spatial waves, right? And so we think that cells are responding to a point source, right, or like a localized source of growth factor, for example. And Michiyuki Matsuda, who's kind of like the legend of biosensors, he's the one who named a lot of them after Pokemons. He has this gorgeous paper in 2015 looking at a mouse that has a ERK reporter in it, and just seeing these in vivo these waves of ERK just spreading throughout the epidermis, right? And other people have seen similar things in other tissues, right? So it's very likely there is a structure to the heterogeneity. In cancer, it's likely that structure breaks down and the heterogeneity is there. There's more from the untrained eye, you're seeing heterogeneity in both cases, but one has a structure to it and one is chaotic, right? And that might make a real difference. But we don't know. We haven't fully decoded all this yet. There's not like a good, our best effort we wrote a 2-part review. This is how you know you've worked in the field for too long. If you write a 2-part review on something. But Ram, who I mentioned, he spearheaded this during COVID, writing this big 2-part review on all the things we know about ERK dynamics and how they're structured. We call it a field guide to ERK dynamics, and that's everything we know. So if if you want like a detailed discussion, that's probably I can send it to you. But,
Has the technique been used in any single cell context, like cancer or diabetic pancreas?
When you with single cell, sorry, with which?
Like it is apparently like single cell technique, and it can be in a disease or disorder or even primary culture.
Yeah, I don't think. I can think Eric Sahai has done a bit of work using. He used a glucose biosensor and looked in tumor, like a mouse tumor model. And tumors took up more glucose, as you'd expect. There was heterogeneity, right? As you'd probably expect. Those are, I'd say, the big takeaways from that so far. I think we're still kind of in all these pathways, kind of really early in the stage of, okay, yes, it's heterogeneous, but is there a structure to it? Like I was just saying, right? And how do we recognize those structures? And I think we're still just barely scratching. And it might be it's one of the problems that's just very hard for us as humans to do, because it might be. In a time series, there's a lot of complexity, right? I also had a grad student, Nant, who did a lot of the, I mentioned him, he did some of those oligomycin experiments. In his later years as a PhD, he tried to start developing algorithms. They would take the time series, right, and then categorize them into different, this one's very, because as humans, we can see this one looks very chaotic, right, and this one looks very regular, right, and of course, there's simple things like Fourier transforms and stuff that can kind of pull out some of that, but usually they kind of fall flat with the noisiness of the data that we have here. So he tried really getting into math and developing some algorithms that that really could separate out the different kinds of behavior. And I'll be honest, it seemed to drive him a little bit crazy for a little while. he got in touch with some great mathematicians at UCLA. They had all sorts of ideas, but it's a really challenging problem. Time series can contain a lot of information. You can run machine learning on it until your computer dies. But the insights you can pull out are still fleeting, right? So it's a challenging area.
Machine learning is the thing from yesterday, right?
Yes, exactly, right. Yeah.
So now we're so much more advanced.
Yeah.
Right.
What are we doing? Wait, what do we do now?
Well, we do transformers and whatnot, right?
Okay.
We do tricks.
Yeah. Okay. Yeah. You can do. He was running autoencoders and things like this exactly. to try it was modern ideas to try to find it, but it was hard to find the signal, right?
So I have follow-up questions to Shen.
Yeah.
So not all cells are cancer cells.
Good.
So some cells, for example, in the heart, and the heart is really important for humans too.
Yeah.
When it starts to die. And so the question is, could you imagine doing this also for myocytes from the failing heart and
Yeah, absolutely. Yeah, no, and that's a very interesting area. And it's partly why we built this mouse, right? Which has it's a Cree-driven cassette, so you can put in whatever tissue you want, and then we can start looking. And Don Burrs and people in the pharmacology department have done a ton of cool stuff, calcium imaging in the heart, and so. There is a connection between calcium and AMPK. CAMKK2 is an important activator of AMPK as well, right? So there is crosstalk there. So we haven't really scratched that area of physiology yet.
But I think you're kind of ready now.
We're about ready now. I think we're at the point where
it took a while.
It took a while, but finally here we are. Yeah. All of us, it's like, you've been here 12 years now. When are we going to
it's the same here. I mean, our methods take a while.
Exactly.
Right? It's easy to sketch up what you want to do, and it takes time.
Yeah.
Right?
So you have to build a lot of the infrastructure.
But I think myocytes would be really interesting here.
Yeah, exactly. I agree. And we sort of scratched the surface a little bit. I had this little video that I skipped over with C2, C12 cells. I think that Madura had. Oh no, I took it out of this one. We have done C2, C12s. They get this cute little. They do some fun stuff too, right? I wasn't going to show that. That was after that.
Okay.
That's the second talk after the talk. But yeah.
Okay, Shen again.
What was the mass spec imaging slide?
Oh yeah, you had something like looked like
Is it
mass
like
spec.
mass spec imaging?
Blue,
Something bluish.
yellow.
Oh yeah. Okay, yeah. That's another model. So we built something.
One,
yeah. What we did here was we stained cells. Again, we did one of these kind of cyclic experiments. Here we go. Where what we did was we tried 16 different metabolic conditions for the cells, right? So what plus or minus glucose, plus or minus glutamine, plus or minus amino acids, plus or minus insulin, to try to make, really we're focusing on mTOR at this point because we know that all these things can turn on mTOR. These are mTOR targets. We did the cyclic immunofluorescence with 16 different stains. So we had this big cube of data. And then the last thing we did was right before we fixed the cells is we put in OPP, which is basically it labels newly made proteins, right? And so you can get, because you're only doing it for a short time, you basically get a protein synthesis rate per cell, right? So then what we did was we asked, can we model how much protein synthesis a cell is doing based on mTOR activity or other things that we're measuring about we had things like AKT, we had all these things. We had measured 4BP, ULK1, ACC, all these things phosphorylated to ask, can we predict how much, what's the cell's actual protein production status based on all these things that are supposedly regulators, metabolic regulators of it. And we built some models. Those models, I have some fondness for those models. We can talk more about, if you're interested, this data set's all out there. To do what we did here, we just ran the whole thing through a UMAP, right? So you could say, like, if we're taking all those 16 dimensions, smushing them down to 2, where in this big blob, which comprises all 16 conditions, where does mTOR activity show up most strongly? It's over here at this tip. Where does protein synthesis show up most strongly? Well, it's not localized just to that little tip there, right? It's kind of a little peripheral to it and kind of spread throughout. You know, what other things does it correlate with? What combinations of things does it correlate with? That's what we were looking for with this model, right? And we got to some endpoint, and perhaps the most interesting outcome of it was that even though mTOR targets on their own are actually pretty terrible at, you know, each one by itself is not very predictive. The combination of them, same scale here, is much better, right? So somehow these mTOR targets are very synergistic predictors of protein synthesis, as you'd expect them to be, right? But they're not. But looking at any one of them doesn't tell you much, right? Which is a little concerning, right? Because we usually plot for our favorite, and then that's it.
It's almost like a little microchip.
Yeah, it's always a little microchip, multiple inputs, multiple outputs. Yeah.
So do we have questions online? Okay, so do we have further questions? Really exciting, very vivid seminar. Thank you for your, engaging discussion and your willingness to discuss, right away. You usually don't.
Yeah, that's my favorite way to do it. Yeah.
Okay, cool.
Good. Thanks.
Anything else from anyone? Well, then, thank you so much for your participation.